Hey everyone, how's it going? In this video, we're going to learn all about histamine, and we're going to look at the histamine signaling pathway. So in our first ever video, we established this cell signaling framework. Basically, messenger molecules get produced, then they sometimes get packaged, depending on the messenger molecule, then they get released, go off and produce responses, and these responses eventually get terminated. So today, we're going to look at how histamine, a very important messenger molecule, goes through these 5 steps. Let's get into it. So before we get into the cell signaling pathway, let's first take a look at what histamine is and what it does. So histamine is a messenger molecule that is involved in allergic responses and immune responses. So your body's immune system is designed to keep your body safe and fight off infection, and it responds to things called antigens. So antigen is anything that triggers an immune response. Now, an allergic reaction is basically an overreaction of your immune system to an antigen that is normally harmless, but your body thinks is harmful. So in most people that don't have an allergic reaction, this antigen would just be something normal. But in a person with an allergy, this antigen, for some reason, their body thinks that it's very, very harmful. So when the antigen enters their body, there's an insane immune response, and this can be threatening and dangerous to the person themselves. So for example, if we have a person with a peanut allergy, right? In most cases, a person's able to eat a peanut and they're fine. But in the person with a peanut allergy, their body thinks that the peanut is harmful. And so the peanut is an antigen that triggers a overreaction of an immune response. And the overreaction of an immune response is what we call an allergic reaction. So immune responses and allergic responses, they both involve many messenger molecules to produce these responses, and a big part of these messenger molecules can be attributed to histamine. A lot of histamine's responses are the ones that we see when we're having an allergic reaction or when we're sick, such as inflammation, itchiness, gastric acid production, airway constriction. These responses may seem dangerous and annoying, but when we have an infection, they're very important as they help our body get better and fight off this infection. It's only bad and dangerous when our body does unnecessary allergic reactions for no reason to harmless stimuli, but unfortunately it happens. But histamine is a key part of these immune and allergic responses. So let's take a look at how histamine goes through our 5 steps of the signaling pathway in order for these responses to happen and eventually get terminated. So let's start off by looking at how histamine gets produced. Your body has a bunch of different types of amino acids. They're very essential for our body to function. And you can see here, this is the general structure of an amino acid. So you have an amine group and you have a carboxylic acid group. Now, in the amino in amino acid refers to this amine group, and the acid refers to your carboxylic acid group. So these are the 2 essential components of an amino acid. Now, one of the many amino acids in your body is called L-histidine, and it's shown in your diagram down here. As you can see, L-histidine has the characteristic carboxyl group, which is the acid side, and the amine group, which is the amino side. So it has the amino component and the acid component, therefore it's an amino acid. And L-histidine also has this characteristic ring structure here. This is the backbone of L-histidine, and it's called the imidazole structure, this ring structure over here. Now L-histidine is relevant and important because it is the precursor of histamine. L-histidine can get turned into histamine by an enzyme called histidine decarboxylase. So you can see that in the word histamine, we have amine. That indicates that histamine contains an amine group. However, it doesn't contain a carboxyl group. So if histamine comes from L-histidine, the amino acid, we have to remove the carboxylic acid group while retaining the amine group. We have to turn the amino acid into an amine. Basically, we have to get rid of the acid components and keep the amino components. And that is done with an enzyme called histidine decarboxylase, or HDC for short. Histidine decarboxylase, as the enzyme name suggests, it decarboxylates or removes the carboxyl group from L-histidine. And when it removes this carboxyl group, now we only have our amine group. going from an amino acid to just an amine. And that's how it turns L-histidine into histamine, our desired amine messenger molecule that can produce responses. That's how histamine gets produced. Another thing to note is that histamine still has this imidazole ring structure. This is the backbone of both L-histidine and histamine. Okay, so with that, we've basically covered the first stage of the cell signaling pathway. We saw how histamine can get produced. Basically, your histidine decarboxylase comes and removes a carboxyl group from your L-histidine, turning it from an amino acid into your amine, histamine. So now that histamine is produced, the next step of the signaling pathway is packaging. And remember we mentioned before that this is an optional pathway. It sometimes happens for some messenger molecules, and in other cases it doesn't. But in the case of histamine, it does happen. Histamine is a polar messenger molecule, which means it has the ability to be stored and retained in storage compartments until later on when it's really needed. So before we look at how histamine gets stored, let's look at some different types of cells that can store histamine. Histamine can be stored in neurons and released and used as a neurotransmitter, or it can be stored in basophils. Basophils are a type of white blood cell. Now this makes sense because, as we mentioned before, histamine is a messenger molecule that is commonly used for immune and allergic responses. And since white blood cells are what produce immune responses, it makes sense that white blood cells will store histamine. ECL cells are cells in your stomach, and they help with the production and release of gastric acid. And histamine is also involved in gastric acid release, so that makes sense why histamine is stored in ECL cells. And finally, histamine is stored in mast cells. Mast cells are basically storage cells. They store various different messenger molecules, primarily those that are associated with immune responses, and as we know, histamine is a big part of that, so histamine gets stored in these storage mast cells. Okay, so now we know a couple of different cells in which histamine can be stored. But how is histamine stored in these cells? We're not going to really get into how histamine is stored in basophils or ECL cells. It's a bit more complicated, but we are going to look at how histamine gets stored in neurons and how it gets stored in mast cells. So in this diagram here on the left, you have a diagram of a neuron. And you see these little yellow circles here at the terminal end of your neuron. So this is the terminal end of your neuron. And these yellow circles here represent vesicles. And vesicles are little storage compartments that can hold on to messenger molecules inside them until these messenger molecules are really needed, at which point they can be released out of your neuron and into your synapse. And one of your many neurotransmitters is histamine. So histamine can be stored inside your vesicles, inside your neuron. And on the other hand, here you have a mast cell, which, as we said before, is a storage cell. So it's loaded with these little compartments, similar yellow circles, except these are called granules instead of vesicles. Very similar idea, just basically a different type of name. Vesicles are associated with neurons. Granules are associated with mast cells, but they basically serve the same purpose. They can store messenger molecules until they're really needed, at which point they can get released out of the mast cell and into the extracellular space. So histamine can be stored inside vesicles or inside granules. And in order to store histamine, in order to pump histamine into these vesicles or into these granules, we have a couple of enzymes to help us out. Let's take a look at what these enzymes are and how they work. So this is a zoomed in picture of your mast cell. And as you can see inside your mast cell, you have a granule. Now usually you have multiple granules, but for the sake of clarity, I've only drawn one granule here. And you can see in the membrane of our granule, we have two enzymes. You have ATPase and you have VMAT2. And these two enzymes work hard to help pump histamine into your granule. So what ATPase does is it uses energy from ATP to pump protons into your granule. So now you have positively charged protons inside your granule. So you have an accumulation of positive charge in your granule. Your granule has a lot of positive charge. This creates what's called the proton motive force. Sorry for my bad handwriting, but the proton motive force is produced by pumping all these protons into your granule. Then your VMAT2 enzyme, which is known as an antiport, it makes use of your built-up proton motive force in order to pump histamine into your granule. What it does is it exchanges your positively charged protons for your positively charged histamine, pumping out your positively charged protons and pumping in your positively charged histamine as a replacement. So it pumps out your protons, and so your protons leave, and in its place it pumps in histamine. So you have an accumulation of histamine inside your granules now. And that's how histamine gets pumped inside your granules, by the work of your enzymes ATPase and VMAT2. And basically the same process happens over in your neurons to pump histamine into your vesicles. ATPase pumps in protons, creating proton motor force, then VMAT2 exchanges the protons for histamine, pumps in histamine, and now you've successfully pumped histamine into your vesicle. And this is useful because now that we have histamine packaged in our vesicle, we have a storage of all this histamine so that when we need an immune response, all your histamine can be released out of your granule, out of your mast cell, and into the extracellular space where it can go off and do responses. And that takes us toward the next step of our cell signaling pathway, which is release. So we've completed packaging. Next step is release. How does histamine get released from neurons or from mast cells? So in neurons, right, like we said, you have your neuron, and at the terminal end, you have a bunch of different vesicles with histamine stored inside. The way that release happens is, as an action potential travels down your neuron, it can stimulate release at this terminal end. So it can allow calcium ions to enter via ion channels, and then that stimulates the release of histamine out into your synapse. So the synapse is the gap between a neuron and another neuron, or it's the gap between a neuron and let's say like a muscle or an organ or a tissue, something else, something that has receptors to which histamine can go and bind onto and produce responses. But that gap in the middle is called the synapse, and that's what histamine gets released out into. So action potentials travel down your neuron, cause the influx of calcium, and then your release process happens, which releases histamine out into your synapse, and then it can go onto various receptors on either other neurons or other effector organs and produce responses inside that neuron or inside that effector organ. That's release in neurons. Now let's look at release process inside mast cells. So on the other hand, when you have messenger molecules such as histamine stored in your granules inside your mast cell, instead of action potentials coming along and stimulating the release, that doesn't happen. Instead, what happens is different things can stimulate the release of your messenger molecules. Things such as infections, toxins, chemicals, heat, injury, different antigens entering your body. Anything that triggers immune or allergic responses can trigger something called degranulation. So we know that inside these mast cells, the things that store your messenger molecules are called granules. So as the name suggests, degranulation is when your messenger molecules escape these granules and leave your mast cell. So that can be histamine or whatever other immune-related messenger molecules you have inside your messenger molecules. They can leave your mast cell and go off and bind to receptors to produce various allergic and immune responses. So they get released out into something called the extracellular space, which is basically the space outside your cell. Now, when they are released into your extracellular space, you can have other tissues or organs nearby, as shown by this little yellow thing. So this is an example of a tissue, and your tissue will have receptors to which histamine can go and bind to. So after histamine leaves your mast cell, it can go over and bind to your receptors and produce responses in your tissues. Now, depending on where in the body you are, different types of tissues can be present. So this tissue is variable. It can be lots of different things. And depending on what tissues are present and what receptors are present, the response can vary. So as we said before, histamine can do lots of different things. That's because it can bind to lots of different receptors on lots of different tissues in lots of different parts of the body, producing lots of different responses. So now we've seen how histamine gets released from mast cells or from neurons, but how does this process actually happen? How is histamine able to escape this granule and exit the mast cell? Well, there's actually a 4-step process. Let's break it down one by one. Now, I've already made a video fully dedicated to the release mechanisms of messenger molecules, and I'd really recommend going and watching that video. I'll link it down in the description below, and I should also have a card for it up at the top that should be popping up around here. But basically, release mechanisms are pretty complicated. It's hard to fully explain it in depth in this video, so be sure to check out that video. It'll really make this clear, but we're going to go over it really briefly in this video. But yeah, for a more in-depth explanation, be sure to check out my other video. Basically, this is your mast cell, and inside your mast cell, you have your granule. And you can see that on your granule, you have these little proteins attached to the membrane. You also have proteins attached to the membrane of your mast cell. So the proteins that are attached to the membrane of your granule or your vesicle, if this was a neuron, are known as VAMPs or V snares. You can remember the V is associated with vesicle or granule, which is basically the same as vesicle. And then these proteins are known as SNAPs or T snares. Now these proteins are critical for the release process. And the release process happens with these 4 steps: translocation, tethering, docking and priming, and fusion and exocytosis. So the first step of translocation, basically it involves your granule or your vesicle containing your messenger molecule, in this case histamine, making its way over closer to the membrane of your mast cell or neuron. It translocates its way over closer to the cell membrane. That's translocation. Then comes tethering. Basically, your SNAP and VAMP proteins link up together. They tether up together. They attach, and that's tethering. Docking and priming is basically when now that your SNAP and VAMP proteins have attached together, now they can have further interactions and connections and likely pull the messenger molecule and the granule closer to the cell membrane as it prepares for the final release. So this is like docking and priming. There's more interactions happening between your SNAP and VAMP proteins. So that's docking and priming. And then comes fusion and exocytosis. So fusion is when your granule or vesicle membrane fuses together with your cell membrane, as you can see here. As you can see, the vesicles fuse, the membranes fuse together, and after fusion, exocytosis can happen. Your messenger molecule has the ability to leave the mast cell and exit into the extracellular space, and that is fusion exocytosis. So just to recap, translocation is when your granule or vesicle moves closer to your cell membrane, and then your SNAP and VAMP proteins tether up together, and then increased connections and more interactions happen between your SNAP and VAMP proteins, that is docking and priming, as it prepares for the final steps of membrane fusion and exocytosis of your messenger molecule out into either the synapse if it's a neuron or the extracellular space if it's a mast cell. And that concludes the 3rd step of our signaling pathway of release. So with that, we've gone over the first 3 steps of the histamine signaling pathway. We looked at how histamine gets produced, how it gets stored into mast cells as well as neurons, and how it can get released with the help of SNAP and VAMP proteins through our 4 steps: translocation, tethering, docking and priming, and fusion and exocytosis. Now for our last 2 steps, response and termination, we're actually going to look at them in the second parts of this video, histamine part 2, because it's just too much to go through in one video. So be sure to stay tuned for that if you're interested in learning about histamine's various responses and how histamine gets terminated. But for now, thank you guys so much for watching. I hope you enjoyed the video. If you have any feedback or questions, comments, suggestions, I'm open to hearing out any feedback, and I would really appreciate it. Please leave a comment down below. But other than that, I'll see y'all next time. Take care.